good afternoon, or good evening. Today, we will be presenting our terminal requirement in laboratory management. We are Group 4 of BSMT-1C. I am Richard Mergal, the leader of the group, and today we'll be talking about our clinical laboratory. In particular, its floor plan, its 2D model, the 3D model, in particular, the sections that are found in our laboratory. So first, I'll be talking about the name of our laboratory, which is QualiMed Test Diagnostics Laboratory. You may wonder, where did the name QualiMed Test Diagnostics Laboratory come from? Well, it came from the words quality, medical, and tests. So in particular, our laboratory is focused upon delivering a wide range of diagnostic services to our community or to the local community to promote public health initiatives. So that is why it is named QualiMed Test Diagnostics Laboratory. In particular, our laboratory is a privately owned, freestanding tertiary laboratory which offers a wide range of diagnostic services. It is equipped with advanced instruments for specialized testing, including clinical microscopy, clinical chemistry, hematology, immunology and serology, and microbiology. Now we'll move on to the laboratory's mission and vision.
I'm Liam Augustine. Our mission here at QualiMed Laboratory is to provide accurate, timely, and reliable laboratory services that support disease diagnosis, prevention, and treatment while upholding excellence, integrity, and patient centered care. Our vision is to be a trusted and innovative freestanding tertiary laboratory recognized for its quality service, advanced technology, and contribution to improving public health and medical diagnosis.
Hello, good day everyone. I am Brian James Cuevas, and now we present to you the Qualimed Test Diagnostics Laboratory Organizational Chart. So, in our organizational chart, we assume or we show that we are in a real clinical laboratory setting. So our organizational chart uses a flat organizational structure that emphasizes the collaboration and streamlined communication. So as you can see at the top is the clinical pathologist who oversees the lab and ensures accurate results and keeps everything compliant. Under them is the chief medical technologist who handles day-to-day operations, supervises each section, and maintains quality control. And we can also see in our organizational chart the five section supervisors, including clinical microscopy, clinical chemistry, hematology, immunology, serology, and microbiology. Each supervisor manages a team of medical technologists who run tests and take care of the instruments. We include also the support staff such as the phlebotomists, receptionists, and utility workers that most likely focus in patient handling, specimen processing, and keeping the lab running smoothly. As you can see, we use circles to emphasize or to present the positions of each role of every role in our laboratory. For example, bigger circles in the chart represents higher position like the clinical pathologist, chief medtech, and supervisor. Yeah, that's all, guys.
So since we are working in a healthcare field, we have to ensure the quality of our results. So we have to have the following safety and quality management features, which first is our standard operating procedures, which are the step-by-step guidelines that ensure all lab tasks from pre-analytical to post-analytical are done safely and consistently. Next is proper chemical storage, where chemicals are organized by hazard class with low-risk reagents in storage rooms and higher-risk ones kept in biosafety cabinets or fume hoods to prevent harmful exposure. Next will be the ventilation system, which is also essential because it removes fumes, gases, and aerosols to keep the air clean and safe throughout the lab. We also have the emergency features like emergency showers, eye wash stations, and fire exits, which are essential, especially if we are handling with stuff that can be harmful, of course, to our employees and, of course, to the samples themselves. Next will be the PPE supply room that ensures that gloves, gowns, masks, and goggles are always accessible so anyone handling chemicals or samples can stay protected. And lastly, of course, we have our biological safety, which is maintained by restricting the lab to authorized personnel only. Sections like microbiology also use a decontamination room to prevent cross contamination. And that's the quick overview of our lab's major safety features.
All right. Hello. Hello. All right. So now we'll be moving on to our digital walkthrough in order to talk about briefly each section of our clinical laboratory. So let me share my screen. So first things first, we will be tackling first the entrance of our clinical laboratory. So as soon as you enter our clinical laboratory, you'll be greeted with the reception area. You may be wondering, why is the reception area facing towards the left as compared to facing towards directly? Well, because we wanted our laboratory to be what do you call this, horizontal. So which means the behind the reception is our clinical laboratory. So we wanted our clinical laboratory to be at least one straight hallway to make sure that the efficiency of our laboratory to make sure that our laboratory tests are efficient, and so that we may be able to distribute the specimens easily and also show and distribute our results. So as soon as you enter, you may be greeted with a row of chairs, and to your side is our cashier, which is on the left. Basically, this is where you will pay for the services that you will inquire in our laboratory. And on the right is where they receive and they give out the results of your testing. Basically, receiving, which I mean is basically where, like, when you get a urinalysis test, you will have to collect it within the bathroom, and then you will give the specimen here on the right side of the counter, and then they will then distribute this to the example like clinical chemistry or clinical microscopy in order to carry out their tests. The door here over to the right is our specimen collection room. And the specimen collection room, this is where we collect our blood, basically through phlebotomy. So in this room, in specifically, there is a sink and also there is a sharps bin. Basically, after you collect blood from the patient, you then dispose of the needle or basically the syringe that you use for phlebotomy inside the sharps bin in order for you to maintain or reduce the risk of possible contamination or infection from the process of phlebotomy. And over through that hole over there is our public bathroom. Basically, we also made it that our public bathroom is only a PDWD type of bathroom. This is in order to, what do you call this? To not discriminate against any genders, to make sure that the phlebotomist or the person who is trying to uh, maintain the bathroom or see what, basically, it was also used in order to see whether or not the bathroom is occupied or not. So basically, it's also the job of the janitor or the staff utility in order to maintain the bathroom. So that is all for this section. We will now move on to the actual sections of the clinical laboratory.
So as we go on, the first section is clinical chemistry section. So this area analyzes body fluids such as blood, serum, plasma, and cerebrospinal fluid, which have various analytical techniques to measure chemical substances that reflects the body's health. So the tests performed in here includes glucose tests, measured blood sugar levels, the kidney functions test, measure waste products to assess kidneys health, hormones test, assess thyroid, adrenal, and pituitary gland, and other tests like protein test, lactate dehydrogenase, creatinine kinase, lipid profile, serum bile acid test, and amylase and lipase. So for our standard operating procedure for pre-analytical phase, where specimens are collected, labeled, and processed. For analytical phase, where the actual testing is done in here, instrument calibration and quality control are performed. So lastly, for post-analytical phase, it involves result review, reporting, and proper documentation.
For the hematology department, this is a specialized laboratory section that studies blood, blood forming organs, and blood related disorders. It examines blood cells, bone marrow, and organs like the lymph nodes, spleen, and thymus. The department analyzes blood and body fluids to assess cell counts and cell appearance and clotting function. Key tests include the complete blood count, which measures major blood components to help diagnose anemia, infections, and blood cancers. Blood smear, which examines cell size, shape, and structure, and coagulation tests such as PT, PTT, and INR, which evaluate the blood's ability to clot and are used to monitor bleeding disorders and anticoagulant therapy. These tests rely on instruments like hematology analyzers, coagulation analyzers, microscopes, and centrifuges, all supported by strict quality assurance. Moving on to the procedures, the department follows a standard operating procedure to ensure safe and accurate specimen handling. Each blood sample receives a unique ID during accessioning for full traceability. During collection, phlebotomists use sterile technique, dispose of needles safely, and monitor the puncture site. The specimen is labeled immediately with complete patient information before leaving the bedside. After labeling, samples are transported to the lab following proper handling rules. Staff follow universal precautions and maintain thorough documentation. Any unlabeled or mismatched specimen is rejected to protect patient safety and testing accuracy. Overall, these practices ensure that hematology specimens are processed consistently and reliably, supporting high quality results and effective patient care.
Okay, so moving on to the immunology and serology section, this area of the laboratory focuses on the immune system, specifically the detection of antigens and antibodies through serum analysis. It plays a major role in diagnosing infections and monitoring immune responses. So some of the key tests performed in these sections include RPR, a rapid plasma reagent, or syphilis test. It is a screening test for syphilis. Next is hepatitis profile. It is used to detect antibodies and antigens for hepatitis A, B, and C. Next is the dengue NS1 antigen test, which identifies the NS1 protein of the dengue virus during the early stage of infection. Last is the HIV screening, which detects antibodies or antigens associated with HIV. Just like other sections, immunology and serology follow standard operating procedures across three phases. The first phase, pre-analytical phase, ensures that test requests are correct and collecting and handling the specimen properly before it reaches the lab. Next, analytical phase, it this is where we log the specimen and follow specific methods and analyze protocols, including quality control to ensure accurate results. Last is the post-analytical phase, where analyzing, validating, and releasing the results, as well as archiving specimens when needed and disposing of them properly.
So for the